Hi everyone. So in the previous video, we talked about this basement detachment step where our cell is starting to detach itself from the basement membrane and move through the matrix. The third step of metastasis is going to be matrix degradation. All right, so there's going to be kind of two different things that come into play when we engage in matrix degradation. It's going to be MMPs and UPAs. So MMPs are going to be if I write down the abbreviation, these are the matrix metalloproteases. And these are going to be degrading the matrix. And in this step, they're produced by a couple different cell types. They're going to be produced by cancer cells. They're going to be produced by cancer-associated fibroblasts. And they're going to be produced by tumor-associated macrophages. So all of these are going to be making MMPs that can then go and degrade the matrix. Which again is kind of that mesh that surrounds these cells when they are outside of the bloodstream. Our second example is these UPAs. And UPA stands for urokinase plasminogen activator. And UPAs are, in particular, they're not going to be produced in an active state. So UPA is going to bind to a UPA receptor, and that's going to induce a cleavage that results in the release of a protein called plasmin. And plasmin is a serine protease. And this is actually the protein that's going to be degrading the matrix. So UPAs are kind of activators of this plasmin protein, but it's plasmin itself that's going to be cleaving the extracellular matrix, and in doing so, it will release growth factors, etc. And it's also responsible for activating MMPs. We're not going to get into detail on how it does that, but those kind of three actions of degrading the matrix, releasing growth factors, and activating MMPs are going to be our three actions of that plasmin protein. And these are going to be degrading the matrix. As our cancer cell moves along through the matrix, one important thing to note is if we have a group of cancer cells, let's say that our blood vessel is over here, and our cancer cell is trying to access that blood vessel, what we're going to see is that our leading proteins are going to have slightly different integrins as compared to our following cancer cells. So our leading cells are going to have different protein expression compared to our following cells. Right? So in particular, our leading edge of our tumor is going to develop a particular integrin, which is called beta 1. And this is going to be present and binding to kind of the matrix components. I'm going to label that MC, along our leading edge of our tumor progression. All right, so we're moving through the matrix. That's our third step of our cancer's progression. And the next step, we'll talk about intravasation and what that's going to look like for these cancer cells.